Hi guys, what we're going to do today is we're going to go through the photosynthesis prep SAC activity. So this has been posted on Edmodo. It's a little different to the other activities that we'll be doing before SACs. Pretty much I just didn't have a practice SAC to give you, so I've had a look at it and analysed it and tried to pick out the key pieces of information and concepts that you'll need to go through to do your SAC. So this needed to be completed at home. We will go through it on a Monday before the SAC as per normal, and then you guys can also watch this video to go through it as well. So the task at hand. So using your understanding of photosynthesis and the below hypothesis, you need to design an experiment. Now, in the exam, you're really going to be hoping for a design question. They're really easy. It just goes through the ditches process that we've been using in Year 11 and now in Year 12 as well. So we'll go through that today. So the hypothesis that you've been given. Now this is really common, and as you can see that ditches there is a H. So they'll either ask you to create a hypothesis based on the results or based on an experiment, or they'll ask you to provide positive results for the experiment that you are designing. Okay, so that's the circumstance that we have here today. So when it comes down to H, you need to ensure that you're outlining results that support the hypothesis that's been provided. So the hypothesis that we have here, photosynthesis occurs at a faster rate in an area with sunlight. Now when you read this, it's really important that you're underlining key words. So what we're looking at is photosynthesis, we're looking how fast that occurs, and we're looking at it being exposed in sunlight. Okay, so just from having a look at that hypothesis, all right, I know for the dependent variable that I need to look at the rate in which photosynthesis is occurring. Okay, the independent variable, so remember that's the one thing that changes. I need to look at the level or even exposure to sunlight. Okay, after we have a look at photosynthesis in more detail, I would like to go through the time period. I think control group and controlled variables, I think that you should have a really good understanding of them now. Pretty much what controlled variables are is anything that remains constant throughout the experiment. So when you are asked to provide controlled variables, it's really important that you're providing ones that are really relevant to the accuracy of the results. For example, it probably doesn't matter how big the beaker is or how big the test tube is for the catalase SAC, as long as we were keeping the reaction, the solutions for the reactions, so the same amount of catalase, same amount of liver, what is the catalase, and then the same amount of hydrogen peroxide. As long as we're keeping those things the exact same, then the size of the beaker is probably really not going to matter so much. As long as they're mixing together, that's what's really important. So keep that in mind when you are asked to go through controlled variables. Now, controlled testing, we're probably not going to see that in photosynthesis, but a lot of you were quite confused. So for C, you've got control group. Control group is just the normal situation. Okay, so we see that in the catalase SAC where there was no catalase present, so it was distilled water. And the reason that we did that is so that we could see what would have occurred without the catalase. Now, a really important part to know that photosynthesis and cellular respiration, they require enzymes. Okay, a lot of things that we talk about in biology require enzymes. Photosynthesis and cellular respiration are quite complex, and they are not going to happen spontaneously. So that means that they are not going to happen without the presence of enzymes. Okay, something as simple as hydrogen peroxide breaking down into water and oxygen will happen by itself. Okay, the presence of the presence of enzymes will increase the time that happens. So like it will reduce the amount of time that it happens in, and that's important for cells because the hydrogen peroxide is quite toxic, and we don't want that to build up in cells. So that's why we have the catalase present to break that down. However, in the enzyme that had distilled water and catalase, that reaction was still occurring. Okay, it was just happening at such a slow rate that we weren't able to monitor it. All right, so that's an important thing to know. Photosynthesis and cellular respiration, like I said, they require enzymes, and they're quite complex, and they're not going to happen without them. All right. So the control testing is pretty much having a look at the results that we are going to get. Sorry, I don't know if I went control group. So control group is the normal. So it has no independent variable. So in the enzyme case, it had no catalase. Okay, so it was the normal situation. It was how that how catalase and how hydrogen peroxide interacted or broke down by itself without the presence of enzymes. Okay, control testing is providing you with information, can be qualitative, can be quantitative, of what reactions look like. So with the catalase SAC, when you were putting catalase and hydrogen peroxide in there together and the enzyme had not been exposed to different temperatures, and you knew exactly how the enzyme was going to react if it was in a working state. So if the active site was not changed, then you would know exactly how catalase was going to react. So you would get the increase of bubbles, you get the pop that you guys were all very excited about, and then you get the splint that was glowing as well. Okay, so that was to tell you or to show you that this is what's going to happen if the enzyme is working, if we're seeing enzymatic activity. Okay, so we're probably not going to look at control testing for our photosynthesis SAC, but it may come up from time to time, more so with things like enzymes. Okay, I'm just going to touch on sample size and repeated measures. So they obviously, the more results that you have, the more accurate your conclusion is going to be. Okay, so it's just like if you guys come out of a maths test and you all go up to each other and you say, well, what'd you get for question 5? And you say, oh my God, 2x squared. And the more people that you speak to and the more people that tell you that you got exactly the same result as them, the better you're going to feel. So the more accurate you're feeling that your results are going to be. All right. So the more times that we see the same result in science, the more accurate that means that our results are. So the more times that we're going to see it, the more reliable it's going to be. Okay. Sample size. So you might be asked to design an experiment on something like metabolism, and you might be looking at, so you need to look at an animal then. Okay. The amount of humans that you could get compared to the amount of monkeys that you could get compared to the amount of rabbits that you could get compared to the amount of mice that you would get would all differ. Okay, so the bigger that I'm just going to write up here. So the bigger the animal is, the more ethics that there are, and also the more cost that there is. Okay, so if you're looking at something like humans or elephants, all right, actually humans, let's get rid of them for a sec because there's a lot of ethics to do with them. All right, if you're looking at elephants, all right, you're probably only going to get maybe 3 to 5. Okay, but if you have rats, or mice, you might be able to have 20 or 30 of them. Okay, one, with the ethics that they feel that the bigger that they are, the bigger brain capacity that they have, and therefore the more intelligence they have, so the more ethics there are, but also the bigger that they are, the more cost that there is. Okay, so try to keep that in mind when you're designing experiments as to how big your sample size is. Okay, so depending on the size of the animal. If we're looking at photosynthesis, I'm really hoping that you guys know that we're looking at plants. The ethics on plants, I don't really think too much about them. They don't feel that they have a nervous system, which they don't, so therefore we can have large quantities of them, and generally they're not too costly as well. Okay, so that's good to keep that in mind. All right, so as I said before, we'll come back to ditches after we've gone through photosynthesis and cellular respiration in a little bit more detail, but I want you to keep these in mind as we're going through them, especially the dependent and independent. All right, so we do have a basic understanding of what we're looking for, but I want you to be more specific. All right, beautiful. Let's move on. So, before we begin, it is very, very, very vital that you are familiar with this below equation. Now, I'm not quite sure what happened to this. It's supposed to be an arrow. It's when I went from Microsoft to my iPad, so I don't know what happened. But you need to know this off the top of your head. You also need to know cellular respiration. I would personally put them onto like a little sticky note or something and put them on the back of your toilet door, on your shower, on your bathroom mirror, depending on how dedicated you are, maybe your lock screen as well. So put it somewhere where you're going to see it all the time. All right, and this is supposed to be, well, these words have all stuffed up, but it's okay. You understand what it says. So this is 6 carbon dioxide, 12 water. You can see it as 6 water as well, but I like to show you it as 12. It makes more sense to me. And then glucose, obviously over here. That's a formula that you need to know now, okay? And you need to know that carbohydrates is 1-to-1 ratio. So therefore, if we're going to have 6 carbon, we're going to have 12 hydrogen, and we're going to have 6 oxygen. All right? So that's glucose and then oxygen. Now, oxygen is a diatomic molecule. Diatomic. Okay, and you know already that the word di means 2. So we always have to see it as a molecule. Okay, has to be bonded together in order for it to be stable. Now, I want you to keep this equation in mind the entire time that we are looking at photosynthesis. I want to just bring you over here to this hopefully familiar diagram that we did in year 11. So if you don't know what this is already, it's a cross-section of a leaf. Okay, now that should be prompting some triggers, hopefully. So a leaf, it's green, as you can see in the diagram, and its main purpose is to absorb the sunlight that the plant is being exposed to. Okay, so I just want to go through this. So A here, we will refer to that as the cuticle. Now the cuticle, we say that on us near our nails, but the cuticle is made up of a wax. So we've got a waxy coat. Now hopefully you've done the trigger here, that wax, it is a lipid. All right, and we all know that lipid is yep, you guessed it, phobic. Okay, so it doesn't like water. So depending on what climate that they live in, and there was a purple page in your year 11 book, depending on how thick this cuticle was, depended on the type of environment that it lived in. So if it was at risk of losing a lot of water, so it was in a high climate, a high humidity or hotter climate, therefore it would have probably smaller leaves to reduce the amount of water that was lost. It also have a thicker cuticle as well, and it probably also have hairs in the top as well to keep the moisture within that, so it wasn't evaporated. Okay, but that's that waxy coat, the cuticle is there to design to reduce the amount of water loss. Okay, so I'll just put that there. It's probably not going to be a question that you're asked, but you know, I think general knowledge is fantastic, and we all love learning about biology anyway, so beautiful. So B here, and then these cells that are along the bottom here as well. All right, you're not asked to label them. However, you need to know that they're the epidermis. Okay, now some of you would have heard of epidermis already, and I really like that word. You probably come across it when you're looking if you're doing health. You'll probably be talking about the epidermis or epidermal layer when you're looking at the development of a fetus. So the epidermis is just the outer layer. So our epidermis is our skin. That's how we know it as. So this is like the skin of the leaf, if you like. Okay. This one up here is called the upper, and this one down here is called the lower epidermis. Okay. Beautiful. Now, the cells that we really want to have a look at. There's 2 categories here. I don't know if you can see that green. So there's this category here of green cells, and then there's this category here of green cells. Now, what can you see is a distinct difference between the 2? So the one up here is obviously more highly dense. So C, more dense, whereas D, there's a lot more spaces in between. Okay, now I'm just going to get rid of this over here. Bear with me. All right, so both these layers here are important for photosynthesis. So they're photosynthetic cells. And remember, that's what we're looking at. We want to find photosynthetic cells in order to go through and do our investigation. Okay, so I'm sure that you've noticed when you've looked at a leaf that there are a top side and a bottom side, and the top side is generally greener and then the bottom side. Okay, the reason being, excuse me, is just the concentration or the density of these cells. So this one here we would refer to as a palisade, potato, mesophyll cells, whereas this one, can you guess it? Spongy mesophyll. Okay, and the gaps in between here are actually the air pockets or the air spaces. Beautiful. The other thing that you might have seen already in year 11 is that you might have seen a structure that looks similar to this, and it's got two sections. I'll give you a hint, we call this the vein. So if you look at this top section, coloured in very appropriately for you there. And then down here, gosh, I'm running out of room again. Sorry, it's a bit hard to see. So the blue there is the xylem, and then the yellow there is the phloem. Okay, so for food. So the living part of this vein is the phloem, okay, and that carries things like sucrose through the plant. All right, it's really important. I'm not quite sure if you guys remember, but the phloem will carry sucrose, not glucose. Does anybody know why? Sucrose is less reactive than glucose. Okay, so if you imagine that there's the cells all in this vein here around the side, and then it would be going across in the other cells, and there's also living cells within the phloem themselves. So can you imagine if you've got like this glucose that's just traveling down along the phloem, and it's in a glucose form? All these cells are going to want to snatch it up. It's too easy for them. They're just going to want to binge on the glucose. Okay, so if it's in a sucrose form, those cells that really need the energy can be bothered pretty much to break down that sucrose into glucose to go through cellular respiration. If it's in that sucrose form, it's only going to those that are really needing the energy source. Okay, the xylem here, hopefully you remember that's that woody, that dead structure that's in a plant, and that carries around the water. Okay, hence why the blue color. All right, so that's important to know. You might also call that the bundle sheath or the vein like we've got here, just so you're aware. All right, the other thing that I haven't gone through that's labeled here on this diagram that's quite important for us is E, and that's nicely put near the air spaces. Actually, it's good. So hopefully that gives you a bit of a clue. So E itself is the stomata. Okay, now I'm just going to bring this down. So stomata are those lip-looking structures that you would have seen from year 11. And they look like that. Okay, so these structures on the outside we refer to as the guard cells. And the structure in the middle we refer to as the stoma. But together, collectively, we call them the stomata. Okay? What you need to know and you need to remember that's quite important, and I'll just change the colour so it stands out that little bit more, is that carbon dioxide is going to be going in, okay, and oxygen is going to be coming out, generally speaking. All right, and as we said before, so these are air spaces. So these air spaces will have your carbon dioxide stores in them. Okay. Now, why it's important that you have storage? Let's go back and we'll have a look at the equation of photosynthesis. Maybe I'll rewrite it over here instead. Actually, that might be a bit easier. Now, you should know this off the top of your head. So while I'm writing this, I suggest that you probably get a piece of paper and you start trying to remember this as well. So you write it down too. All right. So we've got 6 carbon dioxide, 12 water. Now, I don't have a lot of room here, so I'm just going to put a green dot, which represents the chlorophyll. And then I'm going to put a yellow dot at the top, which represents the yellow, just because I'm running out of room. Sorry, did I say sunlight? That's what I meant. And then glucose, which we know has got the 1-to-1 ratio, plus our oxygen. Okay, so when you have a look at this, and I will try and color code this as best as I possibly can. So, water. It's going to be transported through the xylem. Now, where do we get our water from? Well, where do the plants, not us, because we're not plants. We don't photosynthesize. They get it through their root system. Okay? So, let's have a look. Let's get some new colours here. Yeah, that's good enough colour. Carbon dioxide, that's coming in through the stomata. Alright? And remember, there's storages of this carbon dioxide in the air sacs, or the air spaces. That's really important that you know. So you'll notice that even in the palisade mesophyll cells, that they've got some spaces there that does have the ability for carbon dioxide to go in there. Now, why do you think it's important that we do have this storage of carbon dioxide? Because it's a reactant, okay? Now remember, limiting factors. If I have a limited supply of carbon dioxide, then this entire process of photosynthesis is going to be limited based on my limiting of carbon dioxide. All right, so that's why we need to have those storages there that are in there. Okay, moving along, we've got our chlorophyll. We know that that's to do with these photosynthetic cells. So when we are looking at photosynthesis, and I know this sounds really silly, you need to make sure that you've got a tissue or a cell type that has these photosynthetic cells which contain your chloroplast, which has your chlorophyll. Sometimes students get these confused. So chloroplast is the organelle, okay, whereas chlorophyll is the chemical that allows the sunlight to be absorbed. Okay, so that's really important that you know the difference between that. All right, so glucose is then formed. Let's change color again. Let's go for an orangey color maybe. Yeah. So glucose. Now, something that you might not have known before today is that when we write the photosynthetic equation, we usually write glucose. However, there can also be other types of carbohydrates that are produced. So we've already talked about sucrose today, and then we can also talk about starch. All right, so sucrose we know is the unreactive carbohydrate in which it's transported, and can you remember where it's transported? In the phloem. Okay, so in this section here. All right, if those plants desperately need glucose, then they'll just produce the glucose. All right, however, most of the time they'll create sucrose. Starch is generally, can you guys remember where starch is found? Yep, it's found in the potatoes. So anything that's generally below ground is where plants will store their excess energy, all right? And it's in complex carbohydrates, so your polysaccharides, which in this case is starch, all right? So that's generally not made in photosynthesis. It could be. You might be asked that sort of question, but you just need to know that it doesn't always come up with just glucose, all right? And then, of course, we're left with oxygen at the end. What colour have no use? Let's go with red. Okay, your oxygen, and then that is released by the stomata if it's not required for cellular respiration out into the atmosphere. Okay, so this diagram here is quite important for you just to understand how the structure of the plant, in fact, works with the photosynthetic equation. Okay. Remember, you don't have to watch this for the entire time. It takes me a few goes to get all the way through it, so I do not expect you to be watching the entire thing. Just, come back to it or pause it, or just look at specific sections that might have been a little bit more difficult for you when you were going through and doing it. This diagram here that we're going to go through now, you need to know how to draw this. You need to know how to do it in your sleep. All right, so this is a microscopic diagram of, hopefully you've guessed it, a chloroplast. So remember we said that there's chloroplast and then chlorophyll that a plant or a photosynthetic organism requires. So chloroplast is the actual organism itself. Chlorophyll is the molecule or chemical that is required in order to absorb the sunlight. Okay, and remember that sunlight is really important for providing the reaction with energy. Okay, so you've been asked here to draw your own diagram as well as labeling both. So the microscopic diagram as well as the one that you're about to draw. So I'll just move this up a little bit so I can start to draw. So I'm just going to draw it on a different angle. And I really advise that you do draw these in your topic tests, in your SACs, in your exams. So whenever you see a question on photosynthesis, and sometimes you get progressive ones that might be like 3 multiple choice questions, or one question that, a short answer question that's, created 3 different sections in A, B, and C. So draw this. It's not going to take you very long. It's going to get your head in the right space. You're going to, be revising photosynthesis, locations, that sort of thing, structures. I highly recommend you do it. So we've got this section here. Okay, so they're like stacks, and if you've seen the Douchy podcast, he has a great explanation there with an Oreo. So if you like Oreos, maybe go and watch that screencast that he's got up on YouTube, and then you have a good excuse to eat Oreos. Okay, so this section here, it's made up of grana or thylakoid. So they might be interchangeable when you have a look on Google to see different ones. On the outside, so the part that I've done that there, that's called the thylakoid membrane. And remember, all of these are really important that you understand in terms of photosynthesis. So we will go through them in a sec. So that's the thylakoid membrane. The whole stack of the thylakoids or the grana are called the granum. And I know it's a whole heap of unfamiliar words and you might not remember this, but maybe just do it once or twice a week and just try and rote learn this diagram. Before you know it, it is just going to become second nature to you. Okay, on the very inside, so the fluid that's in there, inside the grana or the thylakoid, we refer to that as the lumen. Okay, the general fluid that's out outside of the thylakoid membrane, we refer to that as the stroma. And I think that's pretty much it. That's all you need to know for the chloroplast. So it's not too, in terms of the structures, so it's not too difficult to draw. All right, I would practice going through and having a look at a dodgy microscope diagram. So if you see anything that looks like this, has like lines sort of thing, that's a chloroplast. Okay, the other type of common organelle that you're going to get obviously is going to be a mitochondria, and so you might see like squiggles or something along the lines of this. All right, but you might be given a dodgy diagram. Okay, so expose yourself to them now. So don't give yourself any excuses. You might as well just go out there and learn it now. Yeah, you don't want to come out of the exam being like, oh, I didn't know about that. I'm telling you now, go and do it, because other people in this state are going to. All right, so be proactive with your learning. That's really important. So I'm just going to go through here on the microscopic diagram about the things that we've labelled in ours. So obviously, I'm just going to draw dots here. This is the stroma. Okay, this is a granum, which would be made up of a grana or a thylakoid, and then on that would be the membrane. Okay, and then inside that would obviously be the lumen. Chances are that they're probably not going to get you to label a microscopic diagram. It might pop up in a multiple choice question about what kind of organelles, what kind of function it occurs there, but you need to know it. You need to be exposed to it. Okay. So the next thing that we need to do, there are two stages of photosynthesis. And I trust that you know this already. So I just want to go through this, hold on for a second. So this membrane here, the thylakoid membrane, that's this section here. So when I zoom into it in a second, you'll know. All right, that's the thylakoid membrane. This is the lumen. So the space within the grana, and then out here in the stroma, I've represented here. Okay, before I go into zoom in too much, this section here that happens in the stroma, it's labelled here for you, is the light independent. All right, whereas this one here, that's inside or happens in the lumen and the thylakoid membrane, we refer to, as seen there, as the dependent. Okay, now light dependent and light independent. Break those words down. So dependent means that it relies on light. Okay, independent doesn't require light in order to occur. All right. I don't think that they'll refer to it now as that, but you might be asked a question on it. So I'm going to go through it anyway. This phase used to be referred to as the light stage, and this one used to be referred to as the dark stage. Typical questions that you might be asked. Why is the dark stage reference technically incorrect? So, the light-dependent stage directly requires the light. So when I zoom in, this section here and this section here, they're the chlorophyll that I've been talking about, and they absorb the sunlight. Okay, that starts the entire reaction off because it actually splits the water molecule. Then the protons stay in here, which gives us an electron chemical gradient. The electrons go along, and then they create some ATP for us, as long as boosting up our friends here, which I'll go through in a little bit more detail. So it directly requires that light to get that reaction starting. Okay, the byproducts, so the outputs for the light dependent, are required by the light independent. So the light independent doesn't actually require that light directly. However, if the light dependent doesn't occur and there's no outputs, then there's no inputs for the light independent reaction. Okay, so that's why it's been called the light independent, because it's not dependent specifically on the light. However, saying it occurs in the dark is technically incorrect because it still needs those outputs from the light dependent, which needs the light at the source. Okay? All right, I'm just going to get rid of this. All right, so what we're going to do, now that we've got rid of that, is I just want you to, so this is the activity that we'll do in class, or we have done in class. I'm not quite sure when you're watching this. So I'll bring this over here. So like I've just said, this is the light dependent and this is the light independent. Okay? Now I want you to have a look at, and you're going to get me sick of me saying this, I want you to have a look at the equation again. So remember, it is 6 carbon dioxide plus 12 water, which gives us C6H12O6 plus 6 oxygen. Okay, fairly straightforward. Now, I want you to look at this and I want you to break down your light independent and light dependent reactions. Okay, so like I've said, water is a direct input here. Okay, so I'm just going to write water here. So what happens is the water comes in, and this is in the lumen. Okay, so I'm just going to circle this in a pink, and this is a pink here. All right, so that's stating there that the water is directly required for the light dependent. So water comes in. Okay, so we've got our H2O here. And there's already a lone pair of protons that are hanging out in the chloroplast. Okay, or sorry, the chlorophyll. Even I'm doing it. So what happens is, and these yellow arrows that are there are representing the light that's absorbed by that. So when the light comes along, it actually stimulates the protons. They get really excited, and then they move along. So you can see that arrow there. Okay. Now what happens is every time an electron goes past, there's some protons outside in the stroma. Once they move along, that provides the protein channel with enough energy, and that allows some of those protons to move on in. Okay, the protons, it's really important that you know, are not permeable to this membrane. So that just means that they're not able to get across the membrane. They need a protein channel, and the only way that they're able to move through that protein channel is by having these electrons move along and excite the protein channel, and then they can move into the lumen. Okay, now once these electrons move along, then there's no electrons there, but the chlorophyll really likes to have electrons there, really wants to have some friends. So what it does, it looks around and then it's like, aha, I've seen some water. So what actually happens is the water then splits, and so you can see that it dissociates into oxygen. Okay, so because remember, it's H2O, so it means it has 2 hydrogen, 1 oxygen, and also has electrons in there. So those electrons go up into that chlorophyll, and then that oxygen is actually waste product. Okay, so we don't actually require that oxygen at all. All right, so I'll just write oxygen underneath. So I'll put the inputs up here and then the outputs there, because that's important for you to note. So water is required. The the electrons get excited by the sunlight that's been absorbed into the chlorophyll. This is chlorophyll A. They move along, bring some protons or H ions into the lumen. Okay, they keep moving along. You'll see here that in fact some are moving out. Okay, when those H ions are moving out, what's happening is that they're finding a friend, which we call NADP. Remember, we're looking at photosynthesis, so that's why that's better. That's why it's NADP. Okay, there's another one that we're going to look at for cellular respiration, and he's called NAD. So we need to remember that P stands for photosynthesis. All right, and then he binds with him. So this is what he looked like before. This is when he found a friend. Beautiful. So remember, we don't need that oxygen anymore. That's waste product. We've got rid of that. We split our water. So now there becomes like this high concentration of protons, so the H ions from the water within the cell. And what all things on earth want to do? What's the thing that makes us feel centered, that we can go to sleep at night, makes me feel better anyway? Everything wants to be at equilibrium. Okay, so because we've got this gradient here, and remember, gradient just means difference, so difference in concentration. So we've got a high concentration here, and we've got a low concentration down here in the stroma. Okay, now if you're getting confused as to what I'm talking about, where's going what, please make sure that you have your diagram of your chloroplast in front of you, and you can annotate that as we're going. Okay, so what happens is there's these ATPase. Now this is really important that you know that this ATPase, so important that I'm probably going to write it for you. ATPase. Now you guessed it, ase, it's an enzyme, okay, which means that it is in fact a protein, okay. You might be asked to talk about a particular protein that's used within photosynthesis, okay, and you might be asked to talk about the role in which it has. So what ATPase does is that it takes so if we have a look down here, it's got ADP plus PI, and what it does is once the hydrogen ion moves through from the high to the low concentration, the only reason it goes through that ATPase is because it's permeable to that. Remember, it's not permeable to the membrane, so it moves down through the ATPase, which is a protein, happens to be an enzyme. ADP to PI gives us ATP. Okay, so if you were asked to describe the role of ATPase, you would say something like it would have a role in cleaving a phosphate from an ATP molecule to preserve energy required for photosynthesis. Or you might say that it just creates ATP from ADP, PI that's found in the stroma, and then creates the ATP. Okay, that's very important that you make sure you understand that. So just to clarify so far, light dependent, we required the water, which was split. Okay, then those electrons went around. Pretty much from that we got a high concentration of hydrogen ions in the lumen, which gave us electrochemical gradient. We then came down to our ATPase. Okay, protein, enzyme, creates ATP for us. Okay, when it also comes out of this protein channel, we also get an ATP, PH. Sorry, so the inputs that we've got is water. Okay, then we have ADP plus PI, because remember that's hanging out in the stroma and that's required. And then we have NADP, because it's photosynthesis. Now the outputs that we have is oxygen, because remember when we split the water we don't require those oxygens, they're waste, so they go straight out. And remember, how is oxygen removed from a cell? It's removed through its stroma or stomata. Okay, so the stroma is the hole within the opening within the stomata. So the outputs are water, ATP, because that's been created by the ATPase, and then also an NADPH, because the hydrogen is bonded with that. Okay, so hopefully that's easy enough for you to see. It's very important that for photosynthesis you are able to distinguish the stages, the inputs and the outputs by relating that back to this main equation. And the locations. All right, please make sure that if this doesn't make sense the first time, that you're going over it. So now we're moving along to the light independent, and I'll start using the green for that. So what happens is that remember, we're going to use all of the outputs, or most of the outputs, from the light independent. The oxygen we don't require, that's already left. Probably shouldn't do that, should I? Because that's going to confuse you. Ooh, how do I get rid of that? Just ignore that green around the oxygen, because that's just waste, that's not required. So we're going to take that ATP and NADPH. We're going to take that to the light independent, So, colour I'm using green. All right, have a look at our equation. What have we not used yet? Carbon dioxide. So, that is an input, direct input, for the light independent reaction. What happens is that binds with a 5-carbon molecule, which we can see here, okay, with a carbon dioxide to an enzyme called rubsico. Now remember how I said that all enzymes, the ones that have been discovered quite recently, all end in A's, okay? Some of them do not, and you will just need to remember them by practice, okay? So things like pepsin, which is an enzyme within your stomach, and then rubsico, which is an enzyme in photosynthesis. You just need to remember them, commit them to memory. Okay, so what happens is the rubsico binds with that carbon dioxide, so remember that's an input for the independent reaction, binds with this 5-carbon molecule, and then it becomes quite unstable. All right, what happens is that the ATP and the NADPH, which we can see here, come along, save the day, and they pretty much turn it into 2 stable molecules, which are called triphosphate molecules. All right. Now remember how I said before that carbohydrates, so remember in our typical equation, we are writing glucose, all right? So that's an output for the light independent. But remember, we can have glucose. Hopefully you're beating me to this. Sucrose. Starch. Now I really need you to know the role of these. So this is immediate. Okay, if the cell requires energy immediately, glucose is what we're after. Sucrose is the way in which it is transported through the phloem, okay, for food. That's how I remember it, okay? And then starch is for storage. Generally, it won't be created after photosynthesis because starch is generally put down under the ground, such as things in potatoes, but it's important for you to know, all right? Awesome. All right, let's move on to the next question. I'm not sure if it's on this. Hold on. All right, so I'll just jump back to these questions here. So, what substances should you test? What type of cell, type of tissue? Hopefully you already know that you need to have a plant cell, and of that plant cell you need to find a tissue that has photosynthetic tissue. So, leaves. All right, if you have a carrot, and some kids get this confused, so I'm just going to go through it. It's not a very orange carrot, I know. Okay, carrot. Now, wherever it is green means that it has photosynthetic tissue. All right, it's not going to have, and some kids get this confused even when they're looking at a diagram, a microscopic diagram, and they're asked to distinguish what kind of cell it is. If they don't see any chloroplast, they think, oh, it can't be a plant cell, so they get rid of that. They are not going to, the plant is not going to produce chloroplast if it doesn't have to. All right, it would be a waste for this carrot down here for it to have chloroplast. It's never going to see the sun unless it's being picked and then it's going to be eaten. The only part of the cell, the organism that's going to require to be doing photosynthesis to create the carrot would be for the top bit, the green bit. I'm not even sure what you call that. I don't know if it's called a stem. But that's where the photosynthetic tissue would be, and that's what you need to require, and that would have your chloroplast. The other way you could tell that the carrot itself would be a plant cell is that you would still see a cell wall. You would still see a large vacuole. Okay, so don't be confused if you see a plant cell and it doesn't have a chloroplast. It would be quite mean if they did it to you, but be aware of it. Okay, cool. Next question. How are you going to measure the rate of photosynthesis? Now, I'm just going to jump back to our equation. So measuring the rate, change colour, red. So have a look at your outputs. So you've got glucose or some sort of carbohydrate, and then you've got your oxygen. Okay? Let's talk about this for a second. Okay, these are some photos that I took from a biomacromolecule prac that I did with the year 11s, and then this one over here was diffusion. Okay, so the way in which that you could test for glucose is Benedict solution. So these things, again, are common sense, or general knowledge, should I say, common sense, that you should probably start to build up on. So that's why you're doing all these exam questions and you're working so hard each week so that you build up your knowledge bank, all right? Hopefully you can see here that this 2.1 or 1.2, I think it is, that is in fact a negative, okay? So that's a negative result. That was our control testing, all right? So I put water in here with the Benedict solution. So let's write that here for you. It was water. It was on purpose a negative result. All right, so this is control testing. I know you're all just as excited as I am. Control testing. All right, whereas 1.1 here, I've added the glucose solution. Okay, and this is our positive result. All right, so that's one way you could do it. Like you could, have a look at that because we want to test if there's glucose present. So that you could use Benedict solution. I'm going to zoom in a little bit more here. So what happened is, and hopefully you remember this scenario from year 11, is inside this cellulose tubing, because we did something very similar to Head Start, we've got glucose in here. Okay, and then on the outside we just had distilled water. This is a test strip here. Okay, and this last one here, if we see a change in that color, if it goes like almost like a greeny color, we see the presence of glucose. Okay, so you could use a test strip. Test strip. All right, so if you wanted to go down the track of having a look for your glucose, all right, there's some ways that you could do it. I take that you already know, and again, I'll add this in. So this is testing for starch. So this was exactly the same experiment as this. This was looking at biomacromolecules. So if we have starch and we add iodine. So hopefully you remember this. We did touch on it a lot last year in year 11 and in the Head Start practical as well. So if it doesn't have, so it's got a negative result, therefore we have the yellow colour. All right, so no reactions occurring. That's a little bit hard to see. It's just behind that little white stick there. But if there's a positive reading for starch, it goes like that blue-black colour. Okay, so remember, we could be looking for glucose, we could be looking for starch, we could be looking for sucrose. Okay, sucrose is also a test strip. All right, so again, good knowledge to have. I think that's probably going to be a little bit difficult. Yeah, like it might be a bit temperamental. We might get positive readings from other parts in the cells because there might be already some glucose that's already been in the cell. We might get positive results for that. It might just be a little bit messy, okay? So probably stay away from this when you're talking about measuring the rate of photosynthesis, all right? I just don't think it's as accurate, but it's good to keep in mind, all right? The other thing that we can look at is oxygen. So how have we looked, or how have we been testing for the presence of oxygen so far? All right. So in our last experiment, we used the splint, didn't we? Okay. And we looked at the flame to see how much recognition there was. Okay. How else do you know that oxygen has been present? If you're just making qualitative observations, what else do you see when oxygen is created? Have to think back to junior science. All right. What are some things that you need to see if a chemical reaction is occurring? Oxygen's being made. Bubbles. Okay. So you're saying to me like, oh, maybe we measure the amount of bubbles. Okay, I want you to think a little bit more deeper than that. So what else? We have a look at our equation. What else do we need to make sure that we have provided to our plant? Water. Okay, so go back down. We have to have water. If I put oxygen into water, what's going to happen to that? It's going to float. So remember those air sacs, and remember I said to you that they have carbon dioxide in them, all right? The more photosynthesis that it goes through, the higher concentration of oxygen that we are going to see, okay? So we need to make sure, one, that that plant has enough carbon, because really, that's what we're looking at, all right? You've got to look back at the source of glucose. Hopefully it doesn't freeze on me. You've got to look at glucose, and glucose is C6H12O2. Any carbohydrate is going to have that ratio. All right, they're still going to have CHO. So we need to make sure we've got a source of carbon. All right, because the hydrogen and the oxygen that can get from the water. All right, once it has a large supply of that carbon dioxide, it can go through, it can photosynthesize, and then we're going to have lots of oxygen in those air pockets. Okay, once we have oxygen in these air pockets, so in these ones here, so if we flood those air pockets with carbon, all right, because that's what's important from carbon dioxide, the carbon, not the oxygen, because we can get that from the water. Once we have enough carbon within here, okay, once we start to go through photosynthesis, that's going to be turned more and more, or converted more and more into oxygen. All right, once there's more and more oxygen, if these leaves are in water, what's going to start to happen to the leaves or parts of the leaves, okay, is it's going to start to float because it's going to become less dense than the water. Okay, now if that didn't make too much sense, go back over it, listen to it, ask me to tell you again and again and again. I won't get grumpy, I promise. Okay, so you need to make sure that you've got photosynthetic plant, okay, or tissue from a plant. All right, you need to make sure that you have a carbon source that you can put into that plant. All right, so you might flood the water with carbon dioxide, but you also might want to inject somehow a source of carbon into your leaves. I try looking up bicarb soda, so start to Google that, have a look at that. All right, you've got water, so place the leaves into water. You need to have that as an input. All right, remember our independent and dependent variable is the exposure to light. So one, you might want to have light readily available, one you might not. So you might want to put it in a cupboard or a dark room, okay? And then the oxygen, the way in which you're going to test to see whether that's present is by how it floats, all right? Because the rate of photosynthesis, the faster that that's going to float, I'm probably going to make the assumption that the faster that the reaction is occurring, okay? So if you come up here, so it says to you, how are you going to measure the rate of reaction? We're going to talk about, we're going to infer the amount of oxygen is to the time that it takes to float. All right, the quicker it takes to float, or the less time rather, that means that the rate of photosynthesis is occurring a lot faster because oxygen is being produced faster. Okay, what evidence are you going to prove? What assumptions are you going to make? What results are you going to want to obtain? All right, so you're going to be looking at the time. We're making the assumption that it's the oxygen that's being produced that's allowing for the plant to float. Okay, these questions here, tricky. So the evidence or the proof or the assumptions that we're making, the evidence here for photosynthesis is the time, the rate in which photosynthesis is occurring. The evidence for that is the time that it's taking because the oxygen is being produced. the assumption that we're making about these results is that it is in fact the oxygen that's being produced from photosynthesis is allowing these plants to float. Okay, beautiful. So, next question. Do you have what it takes to make glucose? So we've already started to talk about that. It's got carbon, hydrogen, oxygen. We know that we're going to get our source from maybe bicarb soda. Again, have a look at that on Google. And then our HO we're going to get from our water, because we don't necessarily need the oxygen from carbon, but we definitely need it from the water. Okay, so that's already there. Beautiful. What is the composition of glucose? How is it formed? Where does its well, I don't think that's good English. Where is it obtained from? Okay, so we've gone through that. So hopefully you've got your worksheet in front of you and you're just scribbling all over it at the moment. Okay. There we go. That makes more sense. So the original source of energy, this comes up a lot when we look at food webs, pretty much not so much in year 12, but in year 11. I'm hoping that you remember that one of us would have said to you at least that the main source or the original source of energy in any food web actually comes from sunlight. Okay, you never know, it might come up, just so you know. All right, carbon, remember, bicarb soda, and then hydrogen and oxygen we get from the water. How does photosynthesis always produce glucose? We've talked about that. We've talked about the glucose, the starch, and the sucrose. Okay, what is the role of glucose in organisms? How is it used? All right, please make sure that you're aware that even if we are not photosynthetic organisms, so we're not autotrophs, we still are required to go through cellular respiration. Okay, bacteria have to use external enzymes in order to break down their source of energy so that they can obtain it. So they need ATP just like we do. Okay, so bacteria actually synthesize enzymes through their ribosomes. They excrete it out into their environment. That breaks down the energy, and then the bacteria is able to engulf that. Okay, so cellular respiration. Really important. Okay, now I've gone through photosynthesis in quite a lot of detail. I know that you're probably getting sick of me going through this. So I'm just going to skip on to the activity that we have or did do or doing. I'm not quite sure what you're up to in class. Okay. So again, it's really important, and I'll just dot point this over here before I start scribbling everywhere. So this again, obviously it's a microscopic diagram. Please make sure that you are able to recognize that. Good or bad diagrams, no excuses, be proactive, okay? And then you need to be able to draw your own, something similar to this, okay, and label it, all right? Whenever you talk about cellular respiration, phases of cellular respiration are so important, okay? There's three. There's glycolysis, there's Krebs, and there's electron transport chain. All right. When we have a look at the exam questions at the end of this, it asks you about the 3 stages that's required. Let me just double check the wording on it. It says something about a rabbit. Oh, no, it doesn't. The product of photosynthesis must undergo a 3-stage process for the slug to access the energy in the product. Okay, so just like I was saying before, all organisms need to go through some sort of cellular respiration, whether they have the actual mitochondria or not. So, name and give a brief description of each of these stages. So 3 stages. You need to talk about glycolysis, Krebs cycle, and electron transport chain. Okay, we refer to those 3 stages if we have the mitochondria. If there is no mitochondria, then we just stick with the glycolysis. Okay. But you need to know its location, so we're going to go through that in a sec. Its inputs, its outputs, so exactly what we did for photosynthesis, but exactly the same thing that we're going to do for cellular respiration. And then the amount of ATP. All right, examiners want to know about ATP. That's the whole reason that we go through cellular respiration, so they need you to be specific to it. Okay, so let's have a look. Glycolysis. So I'll just move this over here so I can write inputs, outputs for you. All right, input, output. Remember, you can pause this as any stage. Go get some snacks, go get a drink, go for a sleep, have a shower, whatever it is, okay? So glycolysis, this actually occurs in the cytoplasm, all right? So it doesn't occur within the mitochondria. We actually require as well 2 ATP, so that's an input. And then we need our glucose, and so that's been represented there by 6 carbon. It obviously has a 6 carbon, 12 hydrogen, and the 6 oxygen, but I just haven't put that on there because otherwise it would be a very complicated looking molecule. So I've just simplified it there with its carbon structure. All right, pretty much what happens is it goes through glycolysis. It creates 4 ATP, but because we took 2 ATP to make it, yes, we got 4 ATP, but we refer to that as 2 net ATP. I think it's really important that you include that net ATP. If you write down 4 ATP, pretty sure you're going to be marked wrong. Okay, so make sure that you know that it's 2 net ATP. All right, it's broken down into, so glucose is broken down into 2 molecules. They're called pyruvate. All right, so you get 2 pyruvate molecules. All right, fairly easy. Remember, that happens in the cytoplasm. A lot of students get that confused and think that that's in the mitochondria. All right. We also refer to this as anaerobic respiration. Okay, doesn't require oxygen. So if no oxygen is present, then it's anaerobic respiration. Now, there's an exam question that comes up in your weekly questions. I'm not sure if you've done it yet or not. But it's really interesting to know. So there's different outputs for anaerobic respiration. So if you compare animals and then you compare yeast. So I'm sure that anyone who's worked really hard through like a fitness workout knows that we build up lots of lactic acid, and that can be really painful. Okay, it means that you've worked hard though. Lactic acid. And then some of you may know that yeast is used in things like bread to make alcohol, beer, things like that, and that produces ethanol. So when you have really nice bread, that makes your alcohol, and then when you make really nice bread, you get like those really big holes in the bread, and that's because of the carbon dioxide. Animals do not produce carbon dioxide from anaerobic respiration. That is only yeast, okay? So that's a pointer that you might want to remember as well. All right? So we've gone through the location. It's in the cytoplasm. We've gone through the inputs. We've gone through the outputs. We've gone through the amount of ATP that is produced. All right? So this is the sort of thing that you need to get used to doing. All right. So now we start going into aerobic respiration, which again has the presence of oxygen. Now there's Krebs cycle and then there's electron transport chain. Okay, Krebs cycle must have oxygen there. However, it does not directly require it. Electron transport chain, however, directly requires oxygen and it uses it. So we'll go through that. All right. So you might get asked that. It might be like a really nitty-gritty question, but remember, Krebs cycle requires oxygen to be present but doesn't need it directly, whether it's electron transport chain needs it to be present and needs it directly. Okay, so the outputs that we had from glycolysis was our pyruvate. Okay, now I'm not going to go too much into detail. You can have a look at the class activity, but pretty much what happens is that the pyruvate goes through this cycle, which we refer to as the Krebs cycle or the citric acid cycle, and you can see that the reason why it's called citric acid cycle is because citric acid is within this cycle. Every time a carbon is removed from the pyruvate, it turns itself into carbon dioxide. Okay, so carbon dioxide is formed from when a carbon is removed from the pyruvate molecule, and those carbons don't want to be by themselves. They don't like to be alone, so they want to have 2 oxygens with them. All right, so input, output. Input would be the pyruvate. Pretty much the inputs and outputs are from glycolysis. So you can see exactly similar to what we saw in photosynthesis, that they rely on the outputs from a previous stage for the inputs for them. Okay, so inputs requires pyruvate. What I do want to stress is that you see these little guys here. Okay, they are called, you might see them being called charged carriers or carrier molecules or something like that. It is important that you know their role. Okay, so you might be asked specifically to tell me about the importance or their role or something that they do. These carrier molecules, what do they do within cellular respiration or even in photosynthesis, because we did see them. Okay, we saw, we didn't see FAD, but we saw NADPH, remember, for photosynthesis, but in here we're seeing FAD and FADH. Okay, so you might need to name them. All right, so these molecules provide energy or carriers for hydrogen ions from one stage to the next. It is very important that you are able to explain their response, their job for photosynthesis and cellular respiration. Okay, you need to know that. All right, so for photosynthesis, you would say that NADPH is important for carrying the energy or the hydrogen ions from the light independent for the light independent phase from the light dependent stage to assist in the formation of glucose. Okay, you need to know that. So going back to Krebs cycle, so the input is definitely pyruvate, and then we have FADH and NADH. NADH. All right, the more you go over this, the easier it is to remember. All right, and then outputs. So carbon is being clinched off, doesn't want to be by itself. What are we getting? How are we getting rid of that? In a source of carbon dioxide. So that is an output. Okay, we also, for each pyruvate molecule, are creating one ATP molecule. Now this is something that kids get confused on a lot. They will say that for each pyruvate molecule we are producing 2 ATP and therefore we're producing 4 ATP. Alright, but that is not the case. For each singular pyruvate molecule, alright, you are making 1 ATP. Alright, remember from glycolysis, so, oh, that's not what I want to do, sorry. Up here, we had 2 pyruvate molecules. Both of those are going to go into Krebs cycle, so therefore we're going to have 1 ATP per pyruvate. Okay, so in total it's going to be 2. All right, so it'll go 2, 2 so far. Okay. And then, oh, sorry, before we move on, another question that you might be asked. So there is a molecule here called acetyl-CoA. Okay, so your pyruvate is broken down into acetyl-CoA. That's the first step of the Krebs cycle. Some people think it's not part of the Krebs cycle, but keep it simple, it is. Okay. If you have a lowered source of glucose and therefore pyruvate, what your body can do is actually transform lipids and proteins into this acetyl-CoA. Okay, so lipids and proteins can still be broken down and can be used in this Krebs cycle. All right. There might be a question on that might come up somewhere along the year, topic tests, exams, so it's important that you understand that. Okay, so when there's a lowered source of pyruvate, which would mean there's a lower source of your glucose, that means that you can break down your lipids and your proteins as well. Beautiful. All right, almost there. So, electron transport chain. This is where we really start to talk business. So this is where I'm already going to tell you 32 ATP are going to be produced. All right, so we know glycolysis is 2 ATP. We know that Krebs is 2 ATP. And now we're coming to the big boys, and electron transport chain is 32. So it goes 2, 2, and then a big jump to 32. All right, so when you go down the orders for cellular respiration. So that's how you can remember that. So 2, 2, 2, or 2, 2, 32. Okay, so what happens is, remember we've got these little carriers, and remember it's important that you know what they do. So they're bringing energy or hydrogen ions between the different stages. All right, so you would say that this, they're bringing it from Krebs cycle to electron transport chain to ensure that we are making ATP. If you talk about photosynthesis, you would say that NADPH is assisting in bringing energy or protons or hydrogen ions to the light independent from the light dependent to create glucose. Do you think I've stressed that point enough? Cool. So we call them, you can call them charged particles, molecules, acceptor molecules. All right, that's what they're called. So pretty much what happens in here is they're broken down. So they get broken down into their original state. Okay, so FAD and then NAD, they're electrons and then they're protons. These protons are using energy, are pumped across into the outer membrane, so across the cristae. All right, so again, that's another structure that you need to know about. So it's happening in the cristae. I don't know if I mentioned that the Krebs cycle is happening in the matrix, but that's where it's happening. Okay, so sorry, electron transport chain occurring on the cristae. So remember, the matrix is the middle, and then it's just moving across the cristae into the towards the outer membrane. Okay, because all these protons, and protons, remember, is just another word for hydrogen ions, are being pushed across the cristae. All right, we're going to have an electrochemical gradient, and remember, we saw this in the light-dependent reaction. All right, and then we saw this exact same protein channel, which is called, who is listening? ATPase. Awesome. And remember, it's a protein, it happens to be an enzyme. It is assisting in the production of ATP from ADP plus PI. Okay, so it's going in this direction. All right, and it's a very, very important process in cellular respiration because it's helping us to make 32 ATP. All right, so let's go through quickly inputs, outputs. All right, so your inputs are going to be your little carriers. All right, so you're going to have your NADH, FADH. And then your outputs, you're going to have 32 ATP. All right, but hopefully you've got the equation in front of you and you're like, but miss, what about the oxygen? You haven't really gone through that. No, I haven't. So remember, this one requires oxygen to be present and it requires it directly. So what's going to happen is that the hydrogen ions, when they come back through, let's change colour, see if you can see this. So when they come back through the ATPase, remember that's a protein, it's an enzyme, and it is building up ADP to PI, so that if it's building up, it's anabolic reaction, it's turning into ATP. So once it comes down back into the matrix, oxygen is present. Now oxygen, if I was going to add oxygen and hydrogen together, what am I going to get? Water. Okay, so if you have a look at your balanced equation, which I don't think I've done, so let's do that now. All right, so your balanced equation for photosynthesis. Oh, cellular respiration. Even I'm getting a bit fatigued. Okay, plus CO2 equals, see if you can beat me. You should know this off the top of your head. Okay, let's color code this appropriately. All right, so we'll go. Glycolysis. Glycolysis requires the glucose directly, which turns it into pyruvate. Okay, that pyruvate is then broken down through the Krebs cycle or the citric acid cycle. All right, and then it produces your carbon dioxide. So that's the main thing that happens there. All right, then we go down to the electron transport chain, and that directly requires oxygen. Okay, that directly requires oxygen because when those hydrogen ions are then pumped back into the matrix down the concentration gradient through that ATPase, it binds with oxygen, which then forms its water. Okay, this 36 ATP, remember, it's broken down into glycolysis, which gives us 2 ATP. All right, Krebs cycle gives us another 2 ATP, and then electron transport chain is 32, which gives us 36 ATP. All right, so you need to know where the inputs of cellular respiration are going to and how the outputs of cellular respiration are required. All right, so hopefully that helps and that makes sense. All right, lucky last thing to go through. There is a question that says create a labeled diagram that represents the link between photosynthesis and cellular respiration. Now, there are lots of diagrams that you can find if you just type in literally link between photosynthesis and cellular respiration. I do, however, would like you to do this before you go and Google it. Try and see what you understand. Mine's going to be really basic, but you can add a lot more to it. Okay, so, what equation am I writing right now? Who can tell me? Hopefully you're saying photosynthesis. And then this equation, what is this? Can anybody tell me? Oh, must have ATP, must be cellular respiration. Okay. So, hopefully you're seeing some trends here. So this is photosynthesis, and I've got 6 carbon, I've got 6 water. The output for cellular respiration, 6 carbon dioxide, 6 waters. Okay. Output for photosynthesis, glucose and oxygen. Input for cellular respiration, glucose and oxygen. Okay, can you see how these are interrelated? They are such important reactions you need to know about, all right? The other thing that you need to start looking at is whether they're catabolic or anabolic. So this is building up, so this is creating glucose. So generally speaking, photosynthesis is going to be creating, so it's going to be anabolic. All right, and we know that because it requires the energy from sunlight. Yeah, so it's taking in energy, it's endergonic. Whereas cellular respiration, it's breaking down that glucose into smaller products and it's producing energy. So this one is a catabolic reaction because it's releasing energy and it's breaking down glucose, so it's an exergonic reaction. Okay, beautiful. Now, last thing I promised, we're just going to go quickly back to SCID, dependent variable. So how are we measuring the rate of photosynthesis? So we're going to be looking at the oxygen that's produced by the time it takes to float. Remember, quantitative observation, so numbers, are very important. And I'm running out of room as to where to write that, but quantitative. The more quantitative data that you can have versus qualitative, the better. So N for numbers. Okay, remember qualitative is description. So the more quantitative data, numbers, so time is the one that we're using, the better. Independent variable is the presence of sunlight. So like I mentioned before, you might have one that's directly in sunlight, one that is not. Okay, your time period. I'm going to go ahead and guess and say that it's probably not going to take you any longer than maybe half an hour to see changes in that, depending on how big your leaf sections are. So you might take hole punches, you might make discs, disc leaves, something like that. Again, I really recommend that you do go and Google photosynthesis pracs, and you might leave them for, I'd say, like 20 minutes, so maximum half an hour. That's your time period. Your control group, the normal, which would be that it's not being exposed to the independent variable, and our independent variable is the sunlight, so it would be in the dark. Sorry, I'm scribbling over the other stuff. Controlled variables, remember, make sure that you're specific to the ones that are relevant. So it would be the amount of carbon that you're producing, so that you're providing the plant, the amount of water, the time period that you're leaving it in, the intensity of light that the results are going to be exposed to. Okay, and remember, H for hypothesis. Tell me what a positive result, so something that's going to support this hypothesis. So if those that are exposed to sunlight, if those that are going to be exposed to sunlight, if they're going to have a faster rate, that's going to prove our hypothesis correct. Okay, and remember the sample size. All right, it's plants. There's less ethics. There's less money because they're not that costly. Okay, so you might have 20 discs each and use the whole class as results, or something along the lines of that. Okay, and remember, controlled variables, you'd use exactly the same amount of, exactly the same type of plant, same age of plant, because they might have a different amount of photosynthetic tissue depending on how old they are. Okay, so I'm really hoping that this has gone through some of the key things that you need to know. Pretty much, if you're a little bit confused as to where to go from here, I need you to, I'm going to try and summarize it. Learn the balanced equations. For that, know where the inputs and the outputs come from. All right? Know the different stages. Know their location. All right? Be able to draw and label organelles. And you should already know anyway, you need to know ditches. It's going to keep coming up. All right. So I hope that that's helped. I will, if I get time, will make a part B that goes to the exam questions at the end. I highly, highly recommend that you go through, in particular, the compensation graph. Because remember how we looked at the relationship between photosynthesis and cellular respiration, that compensation graph tends to come up a lot. So that's on the second page for your exam questions. Be able to go through question 3B. So it goes through and give a brief description of each of these stages. So you need to talk about the name of the different stages, the location, and then the inputs and the outputs. Okay, and then the last question, exam question that I would really like you to make sure you're going over is the last question. That's question 1. It's on the very last page, and it talks about Sally and Toby, about how they're not quite sure which biomacromolecule that is. That comes up so often. Okay, it comes up in topic tests, it comes up in SACs, and it comes up in exams. All right, so last year there was a very similar question to that. So please make sure that you're having a look at those. I will try to make a video, but if I don't, I apologise, and get onto them. Okay. Thanks so much, guys, for your patience and all your hard work. I know that it's definitely going to pay off for you. Okay. You deserve a break now. Thanks, guys.